We're going to take notes on pattern of inheritance. Remember to fill in your outline notes. You can stop, pause, or rewind at any time. There are different methods that chromosomes follow when they're inherited. Last chapter, we went over the process of complete dominance. This chapter will go over codominance, incomplete dominance, polygenic traits, and sex-linked. Complete dominance, this is a review of what we did last chapter, is when one allele is completely dominant over the other. The example Mendel used was yellow seed color. So yellow seed color was dominant, and we used a capital letter to represent a dominant allele. It was dominant to green, so we used the same letter, but lowercase, to represent a recessive allele. There was two genotypes we could use to get yellow seeds. We could have two capitals, or we could have a capital and a lowercase letter, where the dominant trait would overrule the recessive trait. The only way to get a recessive trait is by having two recessive alleles, noted with two lowercase letters. This is an example of how we would fill in a Punnett square to answer the question, what happens if two heterozygous pea plants are crossed? And from this, you can see that three of them would have this dominant trait, and one of them is going to have the recessive trait. Because of this, we say the ratio is 3 to 1. So ratio is a new way of reporting out our answers today. 3 meaning 3 boxes, which stands for 75%, which we used last chapter, and 1 meaning 1 box, which is 25%. Incomplete dominance is new for this chapter. So this is going to show a trait that is in between, so think about incomplete and in between the other two traits. So what actually happens is a third trait becomes visible that was not there before. The example in the book uses is something called a 4 o'clock flower. This is a flower like a rose, a carnation. It's just a name of a type of flower. In 4 o'clock flowers, red is dominant, so we're going to use a capital letter to represent red. And it's dominant over white, so we're going to use a lowercase letter to represent white. But the difference in this that makes this an incomplete dominant trait is that when you have a heterozygous, a capital lowercase letter, you end up with a third color, which is pink, which was not there before. So 2 red alleles gives you red, 2 white alleles gives you white, and then this heterozygous group right here gives you a different third trait that was not there before. Remember, a hybrid is a cross between two opposite traits. So red and white is going to give you this hybrid of red and white, which in this case turns out to be pink. Here's how we're going to use this in biology class. So we have 2 pink flowers with the heterozygous genotype. So when you do your Punnett square, you fill it in. And then we want to know the phenotype ratio. So if I look at this, you have to be careful. Stop and think. I have 2 capital R's in 1 box, and this is going to give me the phenotype of red. I have 2 of the hybrids, and this is going to give me the color of pink. And lastly, this last box, which is 1 box, is going to give me the color white. So my phenotype ratio is 1 to 2 to 1. Let's look at some other types. So remember, phenotype is what it looks like. Genotypes are the letters we use to make the notation. So let's say long tail is our dominant trait, short tail is our recessive trait, and curly tail is the hybrid that's in between the dominant and the recessive trait. Let's do a cross for a long and short pig. So a long pig is going to have 2 capital letters, short pig 2 lowercase letters. When you fill in your Punnett square, you'll notice that all of the offspring have the curly tail. We would write this ratio as 0 to 4 to 0, where there are 4 curly, no long, and no short. Our problem number 2 down here, notice that we have a question about how many, if there was 2,000 piglets born out of a different trait. So in this trait we have curly, so we're going to have a long, a capital, and a lowercase, and the other parent is long, so it's going to be 2 capital L's. So we're going to do a Punnett square. Okay, so if I look at my results, the question is how many curly are there going to be? So here are the ones that are curly, and I notice that that's 50% of the offspring, and I know that 50% of 2,000 is 1,000, so the answer is 1,000. How many are going to be short? There are none that are short, 2 lowercase letters in the Punnett square. How many are going to be long? I notice 2 boxes are long, which is 50%, so that's going to be 1,000 pigs as well. Our third pattern of inheritance is codominance. Notice the prefix co, which means, like as in co-captain, doing equal amounts of work, having equal responsibilities. In this case, both alleles will equally affect the look or phenotype of this organism. A couple differences in notation for codominance is we use capital letters for both traits, and we use 2 different letters. So the interesting is a hybrid. So we're going to use 2 different letters. So let's say we use H and then R. So 2 capital letters, 2 different letters. This would represent a hybrid for this one particular trait. The phenotype is going to end up to be a 1 to 2 to 1, and I'm going to show you how. So in this case, this is a true case for cattle. A red bull is 2 capital R's. The white allele, if you get white and white, is going to give you a white coloring. If you end up with red and white, you end up with both red and white spots equally. So this would turn out to be red, 2 red allele, and red allele turns out to be red. If you inherit a white allele and a white allele, this cattle would be white. If they inherit a red allele and a white allele, notice that they're both capitals, 2 different letters, both capitals. It's going to give you the trait red and the trait white. Each of them will show up equally. The breeders call this a special cow coloring called roan. Let's look at a Punnett square. So if we mate a roan bill and a roan cow, our Punnett square would look like this. Okay. If we look at our phenotypes, we have 2 capital R's, one of those, and remember that gives us the phenotype red. We have 2 of the coloring, which they are called roan. And then we have 1 down here, 1, which gives us the color of white. So there's how you got the ratio of 1 to 2 to 1. This is what a actual cattle of this variety, roan, looks like. You can see it has both colorings in it. Codominant is also found in blood type. Blood type not only has codominant alleles in it, but you can also see it also has a recessive allele. So here, 2 capital letters that are different represents a codominant relationship between the A and the B allele. And over here we have O as a smaller letter, so you know that is going to be recessive. This is what your blood looks like. If you have A blood, you have A proteins on it. B blood, you have B proteins on it. If you have AB blood, you have both A and B blood on your proteins on your blood. And O is actually has no proteins on it. All 4 of those are equally healthy in all human beings. This is how you'd write the genotype for blood type. So blood type A, if you get an A allele from mom and an A allele from dad, you're going to have blood type A. You can also get an A allele from mom and an O allele from dad, in which case the A, because it is dominant, will show up as blood type A. The O allele, which is recessive, would be masked and not show up. For blood type B, you could have two capital B's or the capital B and the recessive O. Blood type AB, this is where the codominance comes in, where you have one allele of A and one allele of B. Type O, notice the two lowercase letters, this is recessive. And as you can see from above here, it's recessive to both the A and the B allele. Let's do a cross with this. So here we have a hybrid A. Remember, hybrid means they're going to have two different alleles. So that's going to be the AO. And the AB blood person is going to have an A allele and a B allele. Looking at our phenotype ratio, I have an A blood here, and I have an A type blood there. So I have two A type bloods. So if you look closely, both of these are designated as blood type A. I also have over here a blood type AB, so that comes in 1. And I have a blood type B down here, which would be a 1 as well. So the ratio here is 2 to 1 to 1. This is a case of multiple alleles because AB blood typing you can inherit as 3 different alleles, A, B, or O. Polygenic inheritance is the way most chromosomes in humans and complex organisms are inherited. Poly is a prefix that means many, and this individual trait is influenced by lots of genes. So it could be 12 genes, 6 genes. So the genes on all 3 of these combine together to give you a single trait. For example, skin color. This was an explanation for why there are so many different varieties of skin color. Height is also another one. Hair. These are all polygenic traits. A polygenic traits, their ratio ends up to be a bell curve, where you have a low extreme down here, another high extreme on the other end has a high point here, and this is where you could find your average of the different traits that you are measuring here, the average of that one trait that you're measuring here. Thank you for listening. That's been Fantastico.